The next type of mutagenesis cause that I'd like to discuss is photoinduced, or that which is caused by high energy light like UVA and UVB. This is the kind of light that exists in very strong sunlight, and so what we're talking about here is the kind of DNA damage that can be caused by sunburns and, if you're exposed to UV light in the lab or something like that. And what we're interested in is where this kind of UV light hits DNA, where there are two pyrimidines right next to each other. I have these figures here that are useful for us, and what they show is a DNA backbone with two thymines that are right next to each other. Now, when you have two pyrimidines right next to each other, the figure doesn't show it great, but I'm going to show you here that there are double bonds in the edge of both of these pyrimidines. And when those double bonds are right next to each other, stacked to each other, and you have a UV light photon that hits that, you end up undergoing what's called a 2,2 cycloaddition. You may remember that from organic chemistry. And when that happens, you actually end up with essentially a cyclobutane ring across the stacking area between these two pyrimidines, or these two thymines in this case. And I've never found a really good figure for this, but this is going to have to do. We end up with what's called a thymine dimer, or more broadly, a pyrimidine dimer, because it can happen with C as well. And what that is, as you can see here, is a situation where two thymines right next to each other are linked to each other, and this causes a kink, deformation of the backbone. And the upshot of this is that, as you might imagine, this is not going to be handled very well during replication. So I'll note here, this thymine dimer cannot be replicated. And if you can't replicate a chromosome, that chromosome is basically dead. So we'll learn later about how you excise and repair these thymidine dimers, along with the other types of mutagenesis that can occur. Okay, let's move on to our third cause of mutagenesis, and this is going to be the topic of chemically induced mutagenesis. And there's a lot of different agents that you might be exposed to in the environment or in the lab that can cause mutations. And we're going to focus on what the book focuses on, and that's mustard agents. Now, these compounds are called mustard agents because they have a smell about them that is sort of like mustard. And let's give an example, such as methane sulfonate. Compounds like this, by the way, are called alkylating agents because what they do is they are able to take methanes and ethanes and things like that and attach them chemically to other compounds. So over here we have the methane sulfonate, and if this compound gets into your body, into your cells, into the nucleus, then we might see a chemical reaction that is caused by this, and you might need a base of some type. Maybe water even could do that. But what we're going to see is a deprotonation of a guanine, and that ends up inducing, again, a tautomerization in which this deprotonated nitrogen is going to collapse to a double bond, and the carbonyl here is going to go and attack the methane group right there, and this sulfonate group is going to be a good leaving group. So the product of this reaction is a tautomerized guanine, and it's also methylated over there. So what starts out as a GC base is going to end up with what we call a methylated guanine, and if that undergoes replication, because of the tautomer form, it will not pair with a C during that replication event, and instead it will pair with a T. And of course that T will end up leading to a transition mutation. Let's make sure we can see that again. A GC base original is going to undergo chemical-induced alkylation, and we end up with a guanine methyl group now attached to that C. We undergo one round of replication, and we end up with that guanine methyl group connected to a T because of the alternate base pairing that we have there. And then the second round of replication will lead to that T being paired with an A. So once again, we see after mutation and 2 rounds of replication, we end up with a G to A transition. So that's one way that an alkylation event can cause mutagenesis, and let's talk about a different one. We can also have a mustard agent end up causing methylation or alkylation at a different position on a guanine. In this case, it's on a nitrogen as part of the purine ring. And when that happens, you end up with a positive charge on that guanine group, and that causes the guanine itself to spontaneously degrade and completely fall apart. And what happens in that case is the guanine ring is now missing, and the ribose simply has an alcohol there, and the cytosine that was paired with that guanine is now unpaired. This is what we call a basic hole. That's the whole part, of course, is going to be right here. And this abasic hole is obviously a problem because we're missing a DNA pair. We sometimes call this an apurinic hole because it's not just G's that it can happen to, but it can happen to A's as well. The 2 purines can disappear. So this is a huge problem, and if we tried to replicate this, it would not have a base there to replicate, and it would cause ultimately severe damage to the chromosome. This also needs to be repaired, and we'll talk about that down the line. So in the 4th and last of the videos covering the causes of mutations and mutagenesis, I'd like to talk about intercalation. And this is where flat aromatic compounds insert between stacked bases. And the classic example of this is a compound you might be familiar with. It's ethidium bromide, which, like I said, is flat and aromatic. This is a compound that we use to treat DNA in agarose gels in order to see them, because this is a fluorescent compound. Now, molecules like this are going to insert, and they do so, as we said, in between bases that are stacked right next to each other. So these intercalating agents get in these bases and they stack beautifully, just like bases do to each other. But the problem is that stretches out the DNA, makes it longer, and interrupts the bases as they stack, and really distorts the DNA itself. So let's make a note that intercalating agents distort DNA structure. Now, because there's essentially extra molecules between bases, the real big deal for intercalation is that it leads to insertions and deletions. And for reasons that may not be hugely obvious at this point, we can say that insertions and deletions are a much bigger deal than transitions and transversions in terms of the damage they do to genetic expression. And that's why we like to wear gloves while we use ethidium bromide, because we don't want to cause mutations in our own genes. So that's where we'll leave it, and we'll move on in the next video with repair of mutations.